All right, it's noon East Coast time. So hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High-Resolution Cryo-Electron Microscopy Program. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. With me today, also from NCCAT, is Ed Eng. Joining from the Pacific Northwest Cryo-EM Center, PNCC, is Sean Mulligan. And from the Stanford SLAT Cryo-EM Center, S2C2, is Michael Schmidt. S2C2 has invited today's speakers, Professor Beth Stadtmueller and Heather Liu, who are joining us from the University of Illinois Urbana-Champaign. And Mike will introduce them properly after a quick update from each of the centers to keep you up to date on the resources we make available at no cost to the research community. For anyone joining us for the first time today, this webinar is a regular event we hold the last Thursday of the month to highlight the methods and tips and tricks that groups are using to collect and interpret the data they can obtain with the help of National Cryo-EM Center resources. You can save the date now for our talk on October 26th. PNCC is hosting Kirill Nadezhin from Columbia University Medical Center, who will give a talk on the structural mechanisms of TRPM7 activation and inhibition revealed by Cryo-EM. And the registration link is already available for that talk on the events page at cryoemcenters.org. And on that page, you can also find recordings of our past talks, as well as information about all of the groups that make up the NIH-funded network of Cryo-EM resources. Also, looking forward to the end of the year, our November talk will actually be moved to the start of December. And instead of a formal seminar, we're going to host a roundtable on interpreting sort of confusing micrographs. So this will be a more informal conversation, and you can even come and share your own images if you'd like feedback and opinions. So look forward to more details and the exact date for that roundtable soon. But back to today for Zoom logistics, please send questions at any time during the talk, but using the Q&A box, not the chat. And we'll save the questions to the end of the talk, and Mike will moderate those. So let's go ahead and get started. First, I'm going to turn the mic over to Ed Eng.
Okay. Hi, my name is Ed. I'm here at NCCAT here in Manhattan. We offer a robust access, screening, and training program, and I wanted to highlight on the training. We're co-hosting an integrative structural biology symposium October 13th. That is oversubscribed, but we're having 2 pre-meeting workshops that is going to be on October 12th. One will be on the Chameleon Block3 vitrification device in conjunction with SPT LabTech, and also a Tundra workshop in conjunction with Thermo Fisher Scientific. And both those workshops will include hands-on demos. And if you would like to learn more, go to nccat.nysbc.org.
Thank you, Ed. I will turn over to Sean.
Hello, everyone. My name is Sean Mulligan. I'm a microscopist at the Pacific Northwest Cryo-EM Center. PNCC has 2 types of proposals: screening access and general access. Screening access allows you to come on site for up to 5 days of sample preparation and screening with staff. This proposal is aimed at low resource institutions. Our other proposal type, general access, awards up to 480 hours of microscope time a year for 2 years. This time can be used for screening or data collection, and approved general access proposals are also eligible for our training workshops, which cover topics from sample preparation through data processing. Approved proposals are also assigned a scientific point of contact, or SPOC, who can help you with questions about sample optimization, data collection strategies, and so on. Our center has an Arctica and 4 Krioses, all with K3 detectors. We have a Falcon 4i on a few of them, and 3 out of 4 of our Krioses have energy filters. For sample preparation, we have VitraBot, Leica EMGB2, VitraJet, and manual plungers. We hope to, if you're interested in applying, our deadline is the 1st of every month. We look forward to hearing more from you. Thank you.
Thank you, Sean. And now I will turn over to Mike.
Hi, I'm Mike Schmidt. I'm one of the co-directors of S2C2, that's at Stanford SLAT Cryo-EM Center. We also have 4 electron microscopes for data collection and another one for screening and training. The equipment we have full range of vitrification equipment, including Vitrobots, Vitrojet, and the Chameleon for sample screening and training. We have on-site training now. We trained about 12 people in the last few months. Just simply go to our website at S2C2, just Google S2C2, and you'll come to our website for more information about the Stanford Slack Cryo-EM Center. Thanks.
All right, and I'll turn it over to you, Mike, to introduce today's speakers.
Thank you. It's my pleasure to introduce Beth Stadtmueller and Heather Liu from University of Illinois, Urbana-Champaign. Beth got her bachelor's degree at the University of Wisconsin and PhD at the University of Utah, and after postdocs at California Institute of Technology, Caltech, is now at University of Illinois. In addition to many other research interests she has, she'll be talking today, and Heather also will be talking about immune molecules that are physically inside your body, but topologically outside. They are mucosal antibodies, and very interesting antibodies that kind of meet the microbiome outside where they can before they enter the body. It's very, very interesting things. I met Beth and Heather at the West Coast Structural Biology workshop, and a small plug for these smaller meetings that are regional, that you learn a lot, and they're very, very good, just like this webinar is. Good for good interactions among people, and you learn a lot. It's my pleasure to introduce them, and their talk today is using Cryo-EM to investigate the assembly, structures, and functions of polymeric antibodies. Beth?
Great, thank you so much, Michael, for that introduction. Just gonna get this rolling here. Okay, so yeah, so we're excited to be here today and tell you about how we've used Cryo-EM to investigate the assembly, structures, and functions of polymeric antibodies. So we're really interested in how vertebrates mediate relationships with the external world. Okay, so as Michael said, we're really thinking about things that are happening in the mucosa. So this is a epithelial cell layer, and this would be perhaps like the gut lumen, right, in an external environment. And there you're going to encounter viruses, bacteria, toxins, fungi, all sorts of self and foreign antigens. And some of these are commensal and some are pathogenic. All right, and so in this environment, we have host cells, proteins, and carbohydrates that help mediate this relationship. All right, and we're specifically interested in how secretory antibodies or mucosal antibodies that are secreted specifically into this environment help us to mediate this these interactions. Really, we're aiming to elucidate these relationships and also to manipulate them for therapeutic benefit. So we're a structural biology lab, so we take a structure-centric approach to this question, but we also use antibody engineering in order to essentially ask better functional questions, so using this structural information, and also to explore therapeutic potential. So secretory antibodies adopt unusual polymeric structures, all right? And until 2020, these were very poorly understood, okay? So this is, again, an epithelial cell layer bordered by the lamina propria on the basolateral side, where we have a plasma cell. and bordered on the apical side where we have the mucosa, which is again going to be like your gut lumen, nasopharynx, your genital tract, and that accounts for more than 400 square meters of surface area in each of us. So we're interested in plasma cells that are secreting these polymeric antibodies, okay? And so an example of this is dimeric immunoglobulin A. All right, so antibodies, of course, all have what we call an FC region and then two Fab arms that are going to let them hang on to antigens. All right, and IgA and also IgM have a unique C-terminal extension called a tailpiece, and this allows them to be assembled as dimers with a protein called the joining chain. Okay, so this is what we call dimeric IgA. It is then recognized by this polymeric Ig receptor, which binds to the IgA and transcytoses it to the apical surface of the cell. And there it is proteolytically cleaved, releasing a complex of the IgA and the PIGR ectodomain, which we call secretory component. All right. And so this is secretory IgA. This is your most abundant antibody. Each of you produces grams of this each day. So a considerable amount of energy going into this process. So we're interested in mucosal functions of IgA, but we're also interested in other polymeric antibodies. So in humans, pentameric IgM assembles in an analogous fashion and is released as secretory IgM, and also the PIGR ectodomain can be released as free SC. Okay, so how do these all function in the mucosa? All right, so if we think about perhaps what's in the mucosa, some bacteria or viruses. These have a number of what we call so-called effector functions, or ways that they're going to mediate interactions with these antigens. All right, so one of the mechanisms is adaptive. So these antibodies can coat or can cross-link antigens, either on a macromolecular scale. Here we've seen bacteria actually cross-linked, but could also cross-link individual antigens on the surface of microbes. We also know that there are innate-like properties of this antibody. So the secretory components can interact with host and microbial proteins and carbohydrates. And we also know that there are host and microbial FC receptors. Those are receptors that can engage the FC of an antibody and elicit downstream functions. Also, these complexes can be taken back up by cells and kind of contribute to the process of antigen presentation and other immune functions. So collectively, for the host, this is promoting pathogen clearance, all right, and also microbial homeostasis. And we're also now starting to realize that these antibodies have a lot of different effects on the microbes themselves as well, all right? And so that's something we're also interested in. We also know that when this system is perturbed, it can lead to, of course, pathogen growth and infection, and also dysbiosis, all right? So irritable bowel diseases, a number of different disease states are associated with defects in this process, okay? So we think about IgA and other polymeric antibodies as sort of a multi-tool, all right? And we're really interested structurally in how they assemble and how each part of the tool is going to support these diverse effector functions. And then we're also interested in engineering different capacities onto this tool for therapeutic benefit. So kind of under this umbrella of how does this multi-tool assemble, how does it work, and can we expand its functions, I'm going to tell you, we're going to talk about two stories really today. So the first is the structures of dimeric IgA, secretory IgA, and I'm going to tell you an aside about teleost fish, IgM. I think for a structural biology community, this is kind of a fascinating story. All right. And I really want to emphasize that Cryo-EM has been really critical for understanding these structures, right? So in 2020, the start of 2020, we didn't have the structures of any polymeric antibodies. And in 2020, structures from my lab, Marissa Matsumoto at Genentech at the time, and Junyu Shao at the University of Peking kind of all came out that year and have really helped us understand these antibodies. I'm also not going to talk about kind of half of our lab, which is engaged in using these structures to engineer alternative antibodies to test function and therapeutic potential. But again, without these cryo-EM structures, that whole kind of new aspect of this field would not be possible. All right, so what I'm going to tell you about is kind of an introduction for Heather's work. And Heather's going to talk about the structures of secretory IgA and complex with a microbial and a host FC receptor. And so Heather is actually defending her PhD 1 week from today, and she will be heading towards S2C2 as a postdoc in Naima Sharaf's lab at Stanford. And so hopefully some of you will see more of her as time goes on. All right, so since I'm going to kind of give the introduction and then let Heather take over, I'm going to start with acknowledgements. Of course, my whole lab at UIUC, which is coming up on 5 years old at the start of 2024. And of course, our funding sources, everything I'm going to talk to you about today has been funded by the NIH. We also are using Cryo-EM to investigate avian antibody structures and their response to avian influenza through an HHMI-funded project. So we collect Cryo-EM data at a number of different places. We have an in-house Glacios. We also collect at Purdue, and we've collected on the Caltech Krios. But I really want to highlight S2C2. So we've had multiple proposals at S2C2, and Heather in particular has really benefited from training and data collection. And of course, thanking Michael for supporting this facility and the invitation to talk, Tet and Nathan for help as microscopy scientists, and Amy and Lisa for helping to kind of get these proposals organized. So really a huge thanks to them. Okay, so I'm going to start by, as I said, talking about the structures of dimeric IgA, secretory IgA, and teleost fish IgM. Okay, and our IgA work that I'm going to talk about has been published, and our fish work will be published shortly, is on bioRxiv. Okay, so just to remind you, dimeric IgA is secreted by plasma cells, and the primary form is dimers, okay? It is at least 2 monomers, and this J chain is absolutely required for this process. Humans can assemble higher order structures, such as tetramers and pentamers, okay? So we're really interested in the assembly, how a plasma cell does this, and what this structure looks like, okay? So we targeted the structure of dimeric IgA, all right, and this is one of the first maps that we determined, and I should say that this was at a resolution of about 3.2 angstroms overall. And you can see that we've got these 2 holes, and we don't have any fabs, okay, so the fabs on the structure are disordered. And this was really kind of an exciting moment because we didn't know really the fold of the J chain. We didn't know a lot about this structure, and we were able to build this. And this is, of course, just a representation of how these 4 chains, A, B, C, and D, are coming together with this magenta joining chain protein, okay, to create this remarkably bent dimeric antibody. All right, so there were a number of things that kind of surprised us about this. So of course the J chain kind of playing a dominant role lassoing these 2 antibodies together. We'll talk more about that in a second. But also this bend and how the J chain kind of associates uniquely with each FC. It's also contributing some asymmetry to this complex. And functionally for an antibody, that's unique, right? Because when we think about monomeric antibodies, such as monomeric IgA, which is the predominant form in the serum, we really think about both heavy chains and both light chains as being kind of functionally equivalent. Receptors could engage both sides. The Fabs have kind of even distribution to interact with antigen, all right? And this is the crystal structure of just the monomeric FC alpha, solved in Pamela Bjorkman's lab by Andrew Herr about 20 years ago. And so this is, of course, very different because we're seeing something that's asymmetric, okay? And if we look at that closer, let's see, this movie is not playing. Okay, there we go. you can kind of see how this antibody comes together. So these tail pieces are forming beta sheets that are folding together with strands from the J chain. So we kind of have two sheets, and then we have these two, what we call wings, that kind of grab onto each FC, okay? And so really through this structure, we're seeing that the J chain is kind of playing a obviously structurally critical role in assembly. And as I said, also contributing to the asymmetry of this molecule. All right, so if we look at that a little more closely, I just want to point out that each of these tail pieces interacts uniquely with the J chain, okay? So we, again, 4 identical heavy chains in sequence interacting with this one unique sequence of the J chain, and I'm not going to go into all the details, but each of these forms a unique interaction. And in addition, then we have these 2 wings, all right? And these are each chemically diverse, okay? So they're unique, and they bind different but overlapping sites on each of these 2 FCs, so chain A and chain C, okay? And so overall, this antibody then is a bit different than that it's long been thought to be. So we see that, again, that it is bent, it's relatively rigid in its core, okay, with that J chain kind of folding together with those tail pieces. And so, as I said, for IgA, J chain is required for dimerization or higher polymer assembly, okay? But as it turns out, so in some species, the J chain is not required for this process. And in fact, even in humans, IgM can assemble into hexamers without the joining chain. And so in terms of understanding how this process works, in terms of assembly and what kind of structures we can get out of this process, we were especially interested in teleost fish. Okay, so teleost fish have actually lost the J chain from their genomes. Okay, so their plasma cells can secrete polymeric IgM and an IgA ortholog called IgT, which have been presumed to be tetrameric. And it's bound by a polymeric Ig receptor, albeit with a different structure. And is transported to the mucosa and can become secretory IgM and IgT. All right, so Iris Liu in my lab, a grad student, has really tackled this question of how do these J-chainless polymers assemble? What do they look like? And then, of course, we're thinking about how those function and comparing those to the mammalian antibodies. And so I'm going to go over this really quickly because we don't have a lot of time, but we determined this structure to a resolution of just about 3 angstroms overall. And here's the structure over here. Okay, so this you can see indeed it is a tetramer. All right, so we have these 4 FCs. All right, and then we have this tailpiece assembly in the middle. Okay, so this is of course, quite different because we don't have the J chain, all right, and we do have tail pieces, but instead of kind of folding in parallel beta sheets with the J chain, they're actually forming these alternating structures, okay, that are resulting in this assembly on one side of the structure. And I also just want to point out, although you can probably see that there's a twofold axis of symmetry running through the center of this, where we could fold this in half. If we kind of think about each of these as a separate antibody, there is asymmetry related to, of course, these 2 copies and these 2 copies. So, right, potentially different accessibility for FC receptor, different trajectories of the Fabs. And so we're really, interested in how it is that this fish is able to assemble this structure. Okay, and so if we look closely at this, you'll see it's relatively planar compared to IgA, which is actually bent and tilted. And then if we look at this tailpiece assembly, hopefully you can see that there are 2 chains that are forming these kind of hairpin structures that are going over the top of this kind of beta sandwich in the middle. All right, it's almost a beta barrel actually. And so how is that happening when you have 8 identical heavy chains? And so what we found is that two of these chains, which I'm going to show in dark blue versus light blue, actually fold differently to accommodate this arrangement. So here we have, looking at the dark blue and the light blue, is what we call an A-type and a B-type fold. Okay, and so if we align those, what you might notice is this tailpiece strand is in a different orientation, but also this loop and the residue identity in the G strand and then the following loops in the tailpiece is actually different in these two folds. All right, so it's not really a conformational difference, but actually a folding difference in 2 of the 8 copies of this heavy chain that allow this to assemble this way. And so this is kind of interesting because it's sort of functionally analogous to what the J chain is doing, right? To fold this in 2 different ways, okay? But again, the outcome is quite different, right? With the J chain inducing this bend and some amount of tilt versus this structure, okay? So this kind of shows us that evolutionarily, we've come up with multiple ways to assemble these structures. So, of course, when we think about this, the next step in both of these cases, having produced these antibodies, is taking them to the mucosa and thinking about really, kind of the business end, where these function. And here, in the interest in time, I'm going to focus on our mammalian system. All right, and just tell you that the polymeric Ig receptor, which is going to bind to dimeric IgA, exists in its unligated form in this closed conformation where we have 5 Ig-like domains, all right, that are closed by an interface between D1 and 4 and 5, all right, we know that this work I did as a postdoc in Pamela Bjorkman's lab demonstrated that this would change conformation upon binding to IgA, but we didn't know how, or we didn't know the final structure, okay? And I just want to remind you that the cleaved PEGR is called secretory component, okay? So, of course, we went after the overall structure of secretory IgA, and just point out this was done by graduate student Sonia Kumar-Bharatkar and former postdoc Ben Parker. And here's the map, all right? This is also right around 3.2 angstroms overall. And hopefully you can see it looks similar to the dimeric IgA. However, it's got this large protrusion on the side, okay? And this was, again, one of those really moments when you were looking at a map trying to figure out what was going on. Because SC was long thought to kind of wrap around the core of the antibody and protect it from proteolysis, okay? But instead, when we built this, what we see is that the secretory component, which I've now kind of just added to this schematic, is bound to really just one side of this IgA, okay? So again, still this bent conformation, and then if we turn this and we look at secretory component, those 5 domains are bound on what we call the front face of the molecule, all right? And so, again, this is really kind of adding asymmetry and potential surfaces to mediate interactions to this antibody, all right? So this is really fascinating to us. I'll just point out that if we compare this to the dimeric IgA structure, our angles of bend are the same, and the tilt is slightly different. So that's just the angle of the 2 FCs relative to each other. So again, the J chain probably conferring most of what we see in this conformation here, but the SC potentially causing more tilt in the orientation between these 2 antibodies. So if we think about how SC interacts with this IgA, again, it's going to start in this closed conformation, and this interface has to break, all right? And so we actually find D1 forming contacts actually with every single other chain in this complex. So it's contacting the tail pieces from this FC and this SC and the J chain, all right? Also, we see it's formed this kind of hydrophobic clamp between D1 and D3, all right, which is, of course, quite different than what we're seeing in its unligated conformation. And so it also appears that the conformation that is essentially conferred by the J chain and the tail pieces forming the dimeric IgA complex is kind of optimal for SC to bind in this this unexpected conformation. Okay. So we wanted to think about the functional significance of this whole structure, really, right? So this bent, why is this bent? Why is SC over here? What are the consequences? As we think about how this might affect some of those effector functions I mentioned earlier, we, of course, were first interested in where our Fabs were, because they're disordered in this structure. All right, so in collaboration with the Monte Khorshid's group and his former grad student Nandan Halloy here at Illinois, we set out to model essentially all possible positions that these Fabs could adopt. All right, and so in doing that, we were able to determine that the core of this structure, that geometry, will actually result in each one of these Fabs having a unique, different set of possible conformations. Okay, and if we kind of look at all of them together with the antigen binding sites specifically shown in yellow, you can see that these Fabs are occupying a site that is kind of towards one side of the molecule. All right, and again, each one has a unique set of positions. So it would appear that the core conformation of this molecule is likely to influence antigen binding. All right, so how might that happen? We think that this geometry might promote high avidity antigen binding. It could provide multiple distances, right, for binding to different types of antigens. And perhaps this is particularly advantageous in the mucosa, where you have to cross-link antigens. And also, there's quite a few carbohydrate antigens, which we would anticipate would be quite flexible. So in addition, this modeling, of course, revealed SC being essentially still sterically exposed, particularly this second domain, okay? And so that's something, the role of secretory component when bound to IgA is very poorly understood, all right? So we're postulating that this plays a role in adhesion and interacting with other factors. And so that's something we're currently exploring, and that this modeling helped us to kind of appreciate. All right. Also, I want to just point out that there are two receptor binding sites. We talked about those FC receptors that can elicit downstream events. And these spaces where the Fabs never occlude the rest of the structure are just kind of left wide open and contain two receptor binding sites. All right, so this was really important because when we think about FC receptor interactions with IgA, most of the studies have focused on monomeric IgA. All right, but this structure revealed what would appear to be two evolutionarily preserved FC receptor binding sites. Okay, and so so that I can give Heather a chance to talk about that FC receptor binding question in more detail, I'm just going to sort of conclude this part of the talk by saying that what these structures have shown us is that plasma cells can assemble these kind of unexpected, seemingly asymmetric polymeric antibodies, right, that are going to promote delivery to the host mucosa, and then confer really remarkable potential to mediate these diverse effector functions. All right, and of course we have evidence that this process can be quite different in other species, right, and with other antibody classes as well. Okay, so part of what we're doing is then working to understand how IgA engages specific antigens. So how does the structure influence host microbe interactions? And so there's three primary pathogens that we're targeting: influenza A, Streptococcus pyogenes or group A Streptococcus, and also C. difficile, right? So GAS causing strep throat, also invasive strep, and then of course C. diff causing a lot of problems with opportunistic infections in the gut. And so in thinking about how this structure engages these, we're looking at how Fabs can interact with them. If the function of D2 can influence how antibodies interact with these, and then how host and microbial receptors are also being engaged in the context of these infections. All right, and so with that, I'm going to kind of turn this over to Heather, who's really going to focus on this third question of how Fc receptors engage this structure. Okay.
Thanks, Beth. Hello, everyone. My name is Heather, and I'm a graduate student here at Beth's lab. Okay. So the story that I'm going to tell you guys about is focused on the Fc receptors, and the following work is accepted at Nature Communications and is in press. So as Beth just mentioned, that the structure that we solved, the secretary IgA structure, it shows that this structure adopts a asymmetric conformation. And based on the modeling of the Fabs, we can see that the two Fc receptor binding sites are still exposed. So we want to investigate more about the structures of those Fc receptors and also what are the possible functions. And there are different Fc receptors. On one side, we have those from the host. So the most well-studied Fc alpha receptor is called CD89, also known as Fc alpha R1. So we know that if the immune complex of IgA binds to CD89, it's going to elicit different elimination processes, such as the produce of cytokines, phagocytosis, and also respiratory burst. And people has already solved the structure of CD89 in complex with monomeric Fc alpha. But it is unclear whether CD9 binding to monomeric IgA versus secretory IgA will induce similar immune functions. So that is unclear to us. And there are also other host F receptors like Fc alpha mu R. We only know that it can bind to both IgA and IgM, but we do not know its exact function. And on the other hand, we have Fc receptors from the bacterial. For example, the SSL7 toxin from Staphylococcus aureus. And the structure of it in complex with monomeric Fc alpha has already been solved. And you can see if we compare the structure to the CD89 complex structure, those two proteins are binding to similar site on the Fc. So that means maybe its function is to inhibit CD89 binding so it can help the bacteria to evade those elimination processes elicited by CD89. And there's another example of the bacteria Fc receptor, which is the M4 protein from Streptococcus pyogenes. And the structure of it is unknown, but people has used some mutagenesis studies to map out its binding site on IgA. And it has been suggested that those binding sites are also similar to the CD89 site. So people has been thinking maybe the function of M4 is also to inhibit CD89 binding. So we want to investigate more on that. The reason why it's important is because M4 is from this bug called Streptococcus pyogenes, and this is a very important human pathogen that is infecting more than 700 million individuals globally each year. And it causes mild syndromes like tonsillitis, strep throat, to very life-threatening diseases like toxic shock and rheumatic fever. So we know that this protein M4 is a member of the M protein family, and it is a surface protein that can bind to all subclasses of human IgA in all its molecular forms, so monomeric and also secretory and also dimeric. And we know that it has a 29 residue IgA binding region closer to the C-terminal end of this protein. And just like all members of the M protein family, it is a coiled coil dimer. We know that it is a very important virulence factor for M4, but we do not know its exact biological function. So it's like sticking out on the bacterial surface, and people has observed its presence under the electron microscope as well. So you can see like those fuzzes on the surface are M proteins. So for my project, the specific aims will be to use Cryo-EM to first characterize the human secretory IgA structure with M4 and also with the human Fc receptor CD9. So I'm going to start with the structure of secretory IgA with M4 first. And the way we made the complex is that we first expressed secretory IgA in HAG293 cells and M4 from E. coli, and then we purify the two proteins separately and then incubate them overnight. So we make sure to add an extra amount of M4 so that the two Fc receptor binding sites are fully saturated. So after that, we put that through SEC, and you can see that the first peak over here should be the complex of secretory IgA with M4, and the second one is just the excess amount of M4. And that is also confirmed by SDS page. So we put the content of the first peak and then put that on a Cryo-EM grid. So for this one, I'm just using the normal quantifoil holy carbon grids. And then we collected the data at S2C2, so it's on TM alpha, which is a Krios. And for our initial data set, you can see that almost all the 2D classes, they adopt a very similar orientation. They all look similar, which is also evident from this distribution plot over here. So that means we have a problem of the preferred orientation. So to overcome that, what we did is we tilt the stage at 25 degrees, and then you can see that we're getting more orientation, like from the front view of this complex. And then what we did is we combined the two data sets, and I processed the data in CryoSpark. So we went on several runs of 2D classification. And in the total, we used around 200K particles to do the ab initio 3D reconstruction. And after that, we did several rounds of non-uniform refinements and get to this final map to around 3.007 angstrom. And because we already have the human secretory IgA structure, I docked that structure to the map, and then I hand-built the M4 part. So here I'm showing you like a closer look of the final structure overlaid with the Cryo-EM map. And again, the Fab region is included in our construct, but because of its disorder, it's not built into the map. So we're seeing 2 FC here, and in magenta is the J chain, which is binding to the 2 FCs asymmetrically, and also the secretory component. And over here, we're seeing the M4, which is indeed a coiled coil, but we're only seeing fragment of it, not the full length, is because the coiled coil dimer has inherent flexibility. So that's why the full length one is not visible in the density over here. And if we take a closer look at the interface, we can see that so first, the helix A of M4 is interacting with this strand, which is the G strand on heavy chain A of the FC. And also the other helix, helix B from M4, is also interacting with the same strand on the heavy chain A. And we also see that the darker green chain, which is the heavy chain B, forming contact with both of the helix A and helix B of M4. And most of the contact that we see are polar interactions. So we're observing a quite extensive binding interface from this complex. And those key residues are also confirmed by SPR and mutagenesis. So you might have already been wondering that I have mentioned that there are 2 Fc receptor binding sites accessible on one secretory IgA. But why are we only seeing one copy of M4 bound? So to answer that question, we docked another copy of M4 to the other FC, which is the FCCD here shown in blue. We dock another copy of that here. And if we zoom in on the interface here, you can see the reason why. So basically the J chain, which is shown magenta here, is clashing with M4. And that is because the J chain is forming asymmetric contact with the two FCs. So that's why it's allowing one M4 bound to this FC, but not this one. So that is the reason why we're seeing the 1-to-1 stoichiometry instead of 2-to-1. So if we think about that in terms of the function of M4, people have been previously suggested that the function of M4 is just to inhibit CDD9 binding. But if that's really the case, wouldn't it be more efficient for M4 to bind to both sides of the IgA so that it can block CDD9 binding on both sides? But that is not the case. And if we think of that in terms of M4 being a surface protein on the GAS surface, so first, because we're only seeing fragment of M4, we wanted to see how that looked with a full-length M4. So we used AlphaFold2 to predict the full-length M4 and align that to our structure. So here is what it looked like with a full-length M4. So the M4 is holding all the secretory IgA it encountered at a very similar orientation on the GAS surface. And some functional implications that we can make from this observation is that first, the Fabs of the IgA, it can bind to different surface antigens on GAS. So maybe this kind of orientation is alter the Fab binding to other surface antigens on GAS. And there are indeed a lot of other different classes of surface antigens aside from the M proteins. And the second implication that we can make is that, so the other Fc receptor binding site is still accessible. Maybe this kind of orientation is trying to alter other Fc receptor binding. For example, the host Fc receptor CDD9. And the third implication that we made is that you can see the SCD2, which is the most accessible domain among the 5 SC domain, is very close to the outer surface of this bacteria. So this kind of orientation maybe can help or alter GAS adhesion or localization in our body. So we want to test out those hypotheses. And we started with the second one, which is whether it will alter the secretory IgA binding to host Fc receptors like CDD9, which is also the second specific aim that I mentioned in the beginning. So our hypothesis is that the M4 bound secretory IgA could sterically allow another copy of CDD9 binding. And that is because if we took the structure of CD9 with Fc alpha and dock that to our structure, you can see that there's nothing that is clashing with this copy of CD9. And that is also sterically accessible if we align the full-length M4 onto this complex. So our second aim here is to characterize the structure of secretory IgA with CD9. So if we see a 2-to-1 stoichiometry instead of a 1-to-1 stoichiometry, that would sort of confirm our hypothesis over here. And so I went on and collected the data of this secretory IgA in complex with CD9. We also did the untilted stage and also a tilted stage. And this data was collected at Purdue Cryo-EM Center. And in the end, we got the structure at 3.2 angstrom. And you can see there are indeed 2 copies of CD9 binding to the same copy of secretory IgA. So that confirmed our hypothesis that the stoichiometry is different for CD9. So CD9 is actually a surface receptor, and it is tethered to the myeloid cell surface using the C-terminal end, which is shown in red spheres. And if we compare our structure, which is secretory IgA in complex with 2 copies of CD9 to the published monomeric IgA with 2 copies of CD9, we can see that even though they can both bind to 2 copies of CD9, the relative orientation of the two CD9 is actually quite different. And that means maybe even though they combine both copies, maybe it's going to elicit different immune responses. So we also compare the binding site for both of the structures, and you can see based on the RMSD here, the binding site is actually quite similar, even though their relative orientation is different. So then we're wondering why the stoichiometry is different for M4 versus CD9. So I'm showing you here the binding site that those two Fc receptors used on the Fc of IgA. So in bright yellow, I'm showing you the shared residues for M4 and CD9. And in pink, I'm showing you the residues interacting with M4. And in orange, the residues interacting with CD9. You can see that they indeed shared a few residues, but the actual binding site is actually quite different. So M4 is using more towards the lower part of the Fc, while CD9 is binding more towards like to the junction of C alpha 2 and C alpha 3. And that also sort of explained why M4 can only bind to one side is because the J chain is using the lower parts to interacting with the Fc. So that is why M4 and CD9 has different stoichiometry when binding to secretory IgA. So with that, I would like to summarize our findings and also to talk to you about some future directions. So first, our secretory IgA with M4 structure reveals an extensive binding site on the IgA. We also see some unique residues that have never been reported before. And we observed this very surprising one-to-one binding stoichiometry, even though that secretory IgA has two accessible Fc receptor binding site. And secondly, our secretory IgA with CD9 structure reveals two copies of CD9 bound, one to each site, and they actually orient differently than when bind to monomeric IgA. So this finding will help us to maybe investigate more on how that infects their different functions when binding to monomeric IgA versus binding to secretory IgA. And when we compare the two structures, we can see that this one reveals one occupied binding site that is sterically accessible for other Fc receptors like CD9. So that means it means that the secretory IgA is orienting the M4 is orienting secretory IgA at very similar orientation on the GAS surface, meaning that the M4 function might not just be to inhibit CD9 binding as previously suggested. So broadly, we think that this work helps us to build understanding of host bacterial interactions in the mucosa. And with that, I would like to thank everyone in our lab and also the very helpful training that I've got from the S2C2 Center and also the wonderful Cryo-EM support there, especially Ted, Amy, and Nathan. And I would like to thank Michael for the very nice introduction and invitation for me and Beth. And also I'd like to thank everyone that have helped us to collect data at the Purdue Cryo-EM facility. And with that, Beth and I are happy to take any questions that you might have.
Thank you, Heather and Beth. I do have a question to start with. The whole theme of this is asymmetry for something that you would have expected to be symmetric. And I can see the interesting part about if you allow the FABs to flop around, they don't occlude one of the sites that you need to the FC binding sites that you need. If you had built a symmetric monomer of some sort, or a symmetric dimer of some sort, would that have would the results have been different? I know this would all be modeling, but can you say that yeah, there is a reason for this asymmetry, and maybe the reason is, in addition to the FC, the FAB parts, that there's some reason that asymmetry might be useful. I think asymmetry is a really cool thing, and so maybe you can talk a bit more about it.
Yeah, so it's a really good question. So if we had sort of built a more linear FC and done this modeling, what would have the result been? And I think we have not done that, I will say. But one thing I'll say is, even though we're thinking just about the Fabs in this modeling, if you start to consider this in the context of a bound antigen, right, that having that asymmetry and always directing that, let's say maybe even a giant antigen, right, in one direction, would preserve access to those FC receptor binding sites, for example, right? So I think in other orientations, could it bind antigens and accomplish some functions? Can you imagine that happening? Yes, right. And I think thinking about it in the context of what it's binding and if it has to cross-link things and then still interact with these receptors, I think that's where this conformation might be quite helpful. And also, though, I think, as I said, it also appears important in the assembly process itself, right? So I think, evolutionarily, it's a little hard to kind of balance the pressure on making this versus its function in the mucosa, right? And so it looks like including this J chain and assembling it in this way is, apparently an efficient way to assemble an antibody. But of course, we also see that the fish can do it in a different way, right? And one of the things we don't know about fish antibodies, we know they have FC receptors. We don't know where they bind or really what their functions are. So we're actually hoping, also in the context of studying the bird system, that starting to be able to compare a lot of these structures will help give us more clues, into why this is important, I guess. And so.
Thank you. We have a couple of questions from the Q&A from Martina.
I can interrupt you for just a second. We are running up to the hour, but Beth and Heather have said they can stick around to answer all the questions, well, as many of the questions we can in a reasonable amount of time. For those of you who have to go, thank you for joining us. We will have the recording available, and we'll keep recording all the questions in case you ask one but have to run away. So thanks for joining us, and let's get back to the Q&A. Thanks for letting me interrupt, Mike. Go ahead.
Oh, no. Martina asks, which SIGA have you used for your studies with M4 and CD89? SIGA1 or SIGA2?
We used SIGA1. So actually the FC region of IgA1 and IgA2 is pretty similar. I think there are only a few residue that is different in the FC region. So yeah, they're pretty similar, but we used IgA1.
Okay, so thank you. And Ankush asks, what if you capture M4 in its native condition with FC interaction and screen some tomograms? Would you expect similar ratios of or different binding forms could be observed in native? Now, I don't know how native you want to go. That is in a complete cell, or maybe we can allow Ankush to elaborate, but go ahead with what you have here.
So we actually tried to do some like in vivo studies of this interaction. What we did is we put a fluorescent protein fused to secretory IgA, then we try to image that on the surface of a whole bacteria. But it's not like a super resolution thing, so we cannot see how many IgA is binding to one M4. It's not like single cell resolution, but we do see the IgA localization on the surface of bacteria. So it is happening in vivo for the bacteria, but we are not able to like quantify the ratio based on like because of the resolution.
Thank you.
Yeah, and I would say, yeah, we'd love to take some tomograms of that interaction and kind of try to look at that ultrastructure. We've kind of just gotten, you know, a system where maybe it could work, but I think there's a lot of unknowns, even about like the density of M4 on the surface, and, you know, kind of when and where it's expressed. And then I don't know how many IgAs you can fit on there. So we certainly have been pursuing studies to try to quantify and better understand that, but we certainly haven't gotten to the point of tomograms. So perhaps in the future, that'll be our next cryo-ET pursuit.
Actually, I
Thank you.
have a question as well. I'll take my microphone that I have. So you used tilted data collection to overcome your preferred orientation issue. Was that the only thing you tried, or the first thing you tried?
I think that's the first thing that we tried, but we also like for other antibodies that we are investigating in our lab, like which has similar issue, we also try like different grid type. So I don't know if I can say that.
Yeah.
Like so we have this antibody that is really sticky, so that is also causing orientation bias. And we tried the normal quantifiable carbon grid, which doesn't look good, actually looks horrible. But we used another, like I think it's called the ultra thin grid, which has a continuous carbon layer on the Cryo-EM grid. And that really helped with the orientation and also the aggregation of the particles. So I would say, like, if anyone has similar issues, I would recommend trying that.
Yeah, I mean, we definitely have orientation bias, and as Heather said, stickiness. So it seems that whether they have fabs on them or not, a lot of times they like to stick sort of fab to fab or FC to FC. And that's always a challenge that we're trying to overcome in data collection.
Actually, as a follow-up, have you played with detergents, or is thin carbon your go-to at the moment? I know this is very like sample to sample, but I enjoy knowing what other people's tips are.
So for my protein, I didn't try detergent, but for the other antibody, we did try detergent, and it didn't work. It actually made it worse. So yeah, I think the ultra thin carbon layer works best for our case.
So there are no open questions right now.
Yeah. In that case, we can wrap up. Thank you for sharing that very cool story. Similar to Mike, I'm like fascinated by the asymmetry, so that's really cool. But your maps are so beautiful. Yeah, thank you everyone for joining us, and we hope we see you again soon.
Yes, thank you so much. And